Hello and good morning, respected medical students and future clinicians. I am Dr. Muzamil Liaqat Ali, Assistant Professor of Biochemistry. Today, we're going to master beta oxidation, the principal pathway for the complete catabolism of the fatty acids. This pathway is absolutely vital because during fasting, starvation, and prolonged exercise, fatty acids become the major source of energy for the body, especially for the cardiac muscles, skeletal muscles, and the liver. Understanding beta oxidation is essential for the grasping of the ketogenesis, energy balance in diabetes, and inherited disorders such as carnitine deficiency and medium chain acyl CoA dehydrogenase deficiency. So let's proceed systematically with the biochemical precision and strong clinical correlation. By the end of this lecture, you should be able to define the beta oxidation and explain why it is called beta oxidation. Describe the transport and activation of the fatty acids. Explain the Carnitine Shuttle in detail. Detail the 4 biochemical steps of the beta oxidation cycle. And finally, to calculate the ATP yield from the typical fatty acid, that is palmitate. So these objectives directly link to the high yield topics in the exams, and clinical scenarios involve hypoglycemia and fatty acid oxidation disorders. So let's start with its introduction, right? Beta oxidation is the oxidative catabolism of the fatty acids that occurs exclusively in the mitochondrial matrix. It produces acetyl-CoA, NADH, and FADH2. Acetyl-CoA then enters the TCA cycle, while NADH and FADH2 feed the electron transport chain for ATP synthesis. This pathway is the major energy source during fasting and is tightly regulated to spare glucose for the brain and RBCs. In clinical practice, defects in the beta oxidation leads to the life-threatening hypoketotic hypoglycemia and cardiomyopathy. The name beta oxidation is derived from the fact that the oxidation occurs at the beta carbon, the second carbon from the carboxylic end. The bond between the alpha and the beta carbon is cleaved, resulting in the sequential removal of the 2 carbon units as acetyl-CoA. This process repeats until the entire fatty acid chain is degraded. This stepwise shortening is the hallmark of the beta oxidation. Now, let's talk about the structure of the fatty acid. A fatty acid consists of a hydrocarbon chain attached to a carboxylic group. The carbon next to the carboxylic group is the alpha carbon, and the next carbon is the beta carbon. The long chain fatty acids, like the palmitate that contains 16 carbon, are the most common substrate for the beta oxidation. While the triglycerides are the storage form of the fats in the adipose tissue that contains 1 glycerol molecule and 3 fatty acids. During fasting, hormone-sensitive lipase metabolize them, releasing free fatty acid into the blood. This slide reminds us that the beta oxidation begins only after lipolysis in the adipose tissue. The overall process of the beta oxidation involves 3 steps, or mainly these steps, right? The very first one is the mobilization from the adipose tissue. The second one is the transport in the blood that is bound to the albumin, right? The third one is the uptake into the target cell. The next is the activation of the fatty acyl-CoA. Then this activated fatty acid, the fatty acyl-CoA, transport into the mitochondria via the Carnitine Shuttle, and then the repeated cycle of the beta oxidation in the mitochondrial matrix. In adipose tissue, hormone-sensitive lipase, activated by the glucagon or the epinephrine, inhibited by the insulin, hydrolyzes triglycerides into free fatty acids and glycerol molecule. Free fatty acids enter the bloodstream and are transported to the metabolically active tissues. The next one is the transport in the blood. Free fatty acids are transported in the blood bound to albumin. They are taken up by mainly the liver, skeletal muscle, and the cardiac muscle, tissues rich in mitochondria. Now, let's talk about the tissues. Those are unable to use the fatty acids. The very first one is the RBCs, that contains no mitochondria, and the nervous tissue, that contains very limited beta oxidation capacity, cannot use fatty acid as a fuel. They depend almost entirely on the glucose. This explains why prolonged fasting can still cause hypoglycemia if hepatic gluconeogenesis is impaired. Now, let's talk about the entry of the fatty acid into the target cells. Fatty acids cannot freely diffuse across the membrane. They require fatty acids transport proteins and fatty acid translocase for uptake into the cell. All enzymes of the beta oxidation are located in the mitochondrial matrix. The mitochondria, also known as the powerhouse. The mitochondria also house the TCA cycle and electron transport chain, allowing seamless integration of acetyl-CoA, NADH, and FADH2 for maximum ATP production. Now, let's talk about the activation of the fatty acids. Before oxidation, fatty acids must be activated to fatty acyl-CoA. The enzyme required, that is the acyl-CoA synthetase, also called thiokinase. So the reaction here is as follows, right? Fatty acid plus CoA plus ATP. That leads to the formation of the fatty acyl-CoA and the production of AMP and PPI, the pyrophosphate molecule. This step occurs on the outer mitochondrial membrane, endoplasmic reticulum, and peroxisomes. It causes the equivalent of the 2 ATP because pyrophosphate is rapidly hydrolyzed. So what about the activation reaction? Fatty acid plus CoA plus ATP. That leads to the formation of the fatty acyl-CoA plus formation of the AMP and PPI, right? This is the energy expensive process, right? Reaction, but necessarily priming step for the beta oxidation. So what about the sites for the activation? The number one site is the outer mitochondrial membrane, the main site for the beta oxidation. The next is the endoplasmic reticulum and the peroxisomes. Peroxisomes have the beta oxidation process for the very long fatty acid chains, right? Now, let's have a look on the Carnitine shuttle. Long-chain fatty acyl-CoA cannot cross the inner mitochondrial membrane. The Carnitine shuttle is the transport system that carries the acyl group across. This is a rate-limiting and highly regulated step in the fatty acid oxidation. Carnitine palmitoyl transferase 1, the very enzyme located on the outer mitochondrial membrane. It transfers the acyl group from the CoA to carnitine, forming acyl carnitine. The CPT-I, that is carnitine palmitoyl transferase 1, is the main regulatory enzyme. It is inhibited by malonyl CoA, that occurs in well-fed state and activated by the glucagon. Carnitine acylcarnitine translocase is in the inner mitochondrial membrane exchanges acylcarnitine, and this is the entering for the free carnitine, that is exiting process, right? That is done by the translocase enzyme. The CPT-II, that is carnitine palmitoyl transferase 2, located on the inner surface of the inner mitochondrial membrane. It transfers the acyl group back to the CoA inside the matrix, regenerating the fatty acyl-CoA and free carnitine, which is again recycled. Once inside the matrix, each cycle of the beta oxidation consists of 4 enzymatic reactions that shorten the fatty acyl-CoA by 2 carbons, releasing 1 acetyl-CoA, 1 NADH molecule, and 1 FADH2 molecule. The cycle repeats until the entire chain is degraded. Now, let's move to the 4 steps involved, right, in this process. The very first step is the dehydrogenation. The enzyme required is the acyl-CoA dehydrogenase, chain length specific isoenzyme, right? So the reaction that undergoes here as acyl-CoA that leads to the formation of the trans-acyl-CoA or enoyl-CoA and the production of the FADH2 molecule. FADH2 donates electrons directly to the electron transport chain, right? The ETF, right? The ubiquinone, and that leads to the formation or that end up to the the electron transport chain and the production of 1.5 ATPs per molecule. The second step, that is the hydration. The enzyme involved is the enoyl-CoA hydratase, adds water across a double bond to form the L-beta hydroxyacyl-CoA. The third step, that is the dehydrogenation. That is the second dehydrogenation process, hydrogenation process involved in the beta oxidation process, right? The enzyme here, that is the beta hydroxyacyl-CoA dehydrogenase, oxidizes the beta hydroxyl group to the keto group, producing the beta ketoacyl-CoA plus NADH molecule that produces the 2.5 ATPs via the electron transport chain. The fourth step, that is thiolysis. The enzyme involved in this process and this step, that is the beta ketothiolase, cleaves the beta ketoacyl-CoA using the CoA molecule, right? Releasing acetyl-CoA and shorten the acyl-CoA chain, right? That is ready for the next cycle, right? So let's have an example of the palmitate-CoA that contains 16 carbon. So that leads to the formation of the palmitate-CoA formation, right? Undergoes 7 cycles of beta oxidation, produces 8 molecules of the acetyl-CoA. So now let's talk about the products of the palmitate or the palmitate oxidation from the 7 cycles. The end products will be the 8 acetyl-CoA molecules, 7 NADH molecules, and the 7 FADH molecules, right? So what about the final ATP yield by the end of this process or the beta oxidation of the palmitate molecule, right? So 8 molecules of acetyl-CoA, 7 NADH molecules, and 7 FADH molecules. These are the gross products or the final products of the beta oxidation of the palmitate. So each molecule of the acetyl-CoA produces the 10 ATP molecule via the TCA cycle plus the electron transport chain. So 10 into 8, that is the 80 ATPs, right? What about the 7 NADH? Each molecule of NADH produces 2.5 ATPs. So 7 into 2.5, that is the 17.5 ATPs. And what about the FADH molecule? That produces 1.5 ATPs per molecule. So 1.5 multiplied by 7 molecules of the FADH2, that is the 10.5 ATP. So the gross total, that is the 108 ATPs, right? So activation costs the equivalent of the 2 ATPs. So net yield from the palmitate, that is the 108 minus 2 ATPs, that is 106 ATPs per cycle or the per molecule of the palmitate, that is the 16 carbon fatty acid, right? So this is an extremely efficient energy source. One palmitate molecule yields more ATP than the complete oxidation of the one glucose molecule. That is around 30 to 32 molecules of ATPs, right? So carnitine deficiency or the CPT-I or the CPT-II defects that leads to the impaired long-chain fatty acid oxidation that causes the muscle weakness, cardiomyopathy, hypoketotic hypoglycemia. So what about the high yield points of the beta oxidation that occurs exclusively in the mitochondrial matrix? Carnitine shuttle is essential for the long chain fatty acids, and each cycle removes 2 carbon as acetyl-CoA molecule. And that is highly efficient energy source during fasting conditions. So to summarize this, fatty acids are oxidized 2 carbons at a time in a sequential manner in the mitochondrial matrix, producing acetyl-CoA that enters the TCA cycle and the NADH and FADH2 molecule that feeds the ETC, electron transport chain. The pathway is tightly integrated with the energy metabolism and is critical during fasting states. So what about the important Viva question that can be asked during the Viva? So why it is called beta oxidation? What is the role of the carnitine? And why can RBCs not oxidize fatty acids? So now you can explain all these questions or answer all these questions by having this concept overview about the beta oxidation, right? So we have now comprehensively covered the beta oxidation from the activation and the carnitine shuttle to the 4 enzymatic steps, ATP calculation, and the clinical disorders. You now understand why fatty acids are such a powerful source of energy and how defects in this pathway cause life-threatening metabolic crisis. In the next lecture, we will discuss the ketone body's metabolism and its integration with the carbohydrate and lipid metabolism, right? Thank you very much for your attention. And if this lecture helped you to clarify the beta oxidation and its clinical relevance, please like the video, share it with your batchmates, and subscribe to the channel. And turn on the notification bell so you never miss the next upcoming lecture, right? So this is Dr. Muzamil Liaqat Ali signing off. Revise the carnitine shuttle and beta oxidation and the related ATP calculation daily. So they are of high yield for the exam and ward rounds. So stay focused, stay precise, keep mastering lipid metabolism along with its integration with the carbohydrate metabolism with passion. See you in the next lecture. Thank you very much.